welcome to structure function of DNA. Let me get my pointer here. I need to get going quick. Let me put my timer on here. 12 minutes. So basically we're looking at what DNA is and what is structure. Again, DNA was known to be chemical in all cells by the end of the 19th century, last century. It has the capacity to store large amounts of genetic information, and it can be copied and passed on from generation to generation. It consists of nucleotides and a sugar phosphate backbone. And so if we take a look at this, this is basically what the DNA looks like, and you need to know basically the nitrogenous base that's made up here, and then how it is arranged over here. And we'll kind of go over that a little bit. There's 4 bases that are found in DNA: thymidine, cytosine, adenine, guanine, and if it's RNA, the uracil replaces the thymine. Okay, Watson and Crick, 2 really funny-looking guys, got very famous by elucidating the structure of DNA, and it's kind of one of those great things in that once we figured it out, it was really not that hard at all. And again, if we look at DNA, the base pairs G and C are like wooden rungs on a rope ladder, and then when we twist that ladder, we get the double helix structure that we know today. The base pairs that we're talking about is that whenever we see an A, it is paired with a T. Whenever we see a C, it is paired with a G, and we'll talk about that bonding in a little bit, and that's critical for that. And so what we see is the bondings that hold the sides of that rung on the ladder together are what we call hydrogen bonds. And so those hydrogen bonds are only going to last up to about 100 degrees. If we look at this structure in this computer-generated model, it is very consistent in size. And so the A is a little bit larger, the T is a little bit smaller, the G is a little bit larger, the C is a little bit smaller. And this bonding occurs between those. And so here's our kind of ladder model. Here is our model that we have, and so it's important for us to kind of know about this. When we look at DNA replication, what's really kind of interesting is that when we make a new strand of DNA, we're not necessarily making a photocopy. What we're doing is we're taking that double-stranded DNA, we're splitting that strand, and then we're adding, due to the base pairing rules, C with G, here C with G, T with A, etc., we're adding that new piece. So whenever we make a new DNA daughter molecule, it's half the old molecule and it's half of a new molecule. And so that's really interesting. The types of enzymes that do these are called DNA polymerase, where it reads DNA and it makes DNA. DNA replication in eukaryotes is pretty complex, and I'm really not going to go into that. But what we do is we have what are basically 2 origins of replications, or 2 replication bubbles that help us make our 2 daughter-stranded. So rather than just read it on one site, you can do many. What I want to get to here is the flow of genetic information from basically DNA to RNA to protein. And there is a specific flow of information that is very important. And so we'll take a look at that. Oh, God. Okay. Sorry about that. And it's the wrong site. So an organism's genotype can determine its phenotype. And so a genotype is the genetic makeup of an organism. The phenotype is what an organism's traits give us. And how this happens is that we see transcription, which is a transferred genetic DNA from DNA to an RNA molecule. And so the DNA is very stable in the nucleus, and what we need to do is we need to convert that DNA into an RNA molecule so that it can go out into the cell. Now, an RNA molecule can be damaged and destroyed, but the DNA will always be constant. That's one of the reasons. What we call translation is transformation information from RNA basically into a protein. It's the primary sequence of a protein. And so here we have DNA. It's in the nucleus, and part of what the nucleus does is it protects that DNA. However, in order for that information to be used, we need to convert that DNA a very small portion of that DNA, a gene-sized portion of that DNA. We need to convert it into a piece that can then move outside the nucleus. And then once it does that, it's through a process of what we call translation, that can be that DNA information can actually be used to make a polypeptide, and that's what's really important. Again, genetic information in your DNA is transcribed into RNA and then translated into proteins. One of the big codes that we cracked when we looked at DNA was basically what is that genetic sequence. This is looking at the bread mold rhizopus that helped us kind of crack this code. But basically what we have in a genetic code is that we have what we call 64 triplets. There's 61 of these triplets that code for amino acids, and 3 are basically stop codons. And so basically what's going on here is that when we have these genes are made up of a sequence, but each one of those sequences, a codon of 3 nucleic acid, makes up 1 amino acid. And the reason why we had to do this is there's only 4 base pairs. There's 20 amino acids. So in order to really cover 20 amino acids, we need to have our words be a little bit larger. And this is exactly what we see. So phenylalanine is coded for by UUU and UUC. AUG is the start site. UA is stop, UAG is stop, and UGA is also stop. And the rest of these code for the amino acids that are out there. DNA is started at the promoter, and we'll talk about that a little bit before, and that's where the RNA polymerase binds and will make a copy of a gene. Genes are very interesting, and so here are naked mole rats, one of which has been cloned with the green fluorescent protein from jellyfish. And so we can take genes are universal. We can take genes from different organisms and put them together. Again, here's RNA polymerase. That RNA polymerase starts at a promoter, and then it makes a copy of the gene, and then basically it goes up to a termination sequence, and then turns off. And so this is what we call an open reading frame, where we have a start to an end, and there's probably going to be a protein in that open reading frame. Translation, I'm not going to get that much into translation, but basically what we do is we have a piece of DNA where we have exons and introns that are kind of all pieced together, and so that our coding sequence comes out into the cytoplasm as an intact structure. RNA ribosome is responsible for converting the basically the primary sequence of a messenger RNA into amino acid, and we'll really talk about that, but you do need to know that it's relatively complex. We see initiation, elongation, and termination just like we did. And so just to review, genetic information is what we call the central dogma, is held within all of our cells in the form of DNA. That DNA is converted to RNA that can go out into the individual cells, and that RNA is converted into protein by that process. And so here's a little bit of what's going on with that process. So it's very interesting. The next thing we're going to look at is mutations, which is basically a change in the nucleotide sequence of DNA. Mutations can change the amino acids in a protein, but they don't necessarily have to. And we'll take a look at that. And so we have a number of different mutations. We have what's called a base substitution, which is a replacement of one base by another. We have a nucleotide deletion, which is the loss of a nucleotide. We have a nucleotide insertion, which is the addition of a nucleotide. Because we have a triplicate codon, whenever we do something that's not part of a triplicate, it can have a great effect. And so in this one here, what we're doing is gene replacement, is if we replace this T with an A, what's going to happen is we're going to go from having a glutamine to a valine, which is basically the actual sequence that we see, single nucleotide polymorphism, that gives us the difference that is malaria. Okay? And mutagenesis can occur in a number of different things. And again, this is just looking at the different types of mutations that are going on. And so here we have the normal gene. When we do a base substitution, we can convert this glycine into serine. All the other amino acids are all exactly the same, though. So this is not that big of a deal. Again, if we delete a base pair, what happens is that triplicate codon, which was here, UUU, goes to UU. Well, we deleted out the U. G, that's another amino acid. So all the amino acids that follow this portion could be deleted in some way. Okay. Again, we look at these structures in here and we see that. And I hope you had a really great night. And again, this is a table looking at the different effects of different mutations.